Hello student, welcome to today's lesson. This is biology lesson for grade 12. Student, in our last lesson, we have seen respiration, some stages of respiration, glycolysis, and then link reaction. Under this lesson, we are going to see Krebs cycle, and again end product, and again oxidative level phosphorylation. At the end of this lesson, student, we will be able to determine end product of Krebs cycle after its regeneration, and again explain the role of chemiosmosis and the oxygen in inner membrane space of mitochondria. Student, what do you think may be happen in Krebs cycle? Well done. In Krebs cycle, there are so many reactions takes place. It is one type of aerobic process that takes place in mitochondria. The reaction takes place in mitochondrial fluid matrix, and again it is aerobic one. Here in Krebs cycle, acetyl-CoA, which came from that of link reaction as a linking between that of glycolysis and that of Krebs cycle, react with 4 carbon compound oxaloacetate to form 6 carbon compound called citrates. Here there is a binding of acetyl-CoA with that of oxaloacetate to form 6 carbon compound citrates. Here there is an enzyme that can catalyze the process. Acetyl-CoA from that of link reaction plus that of oxaloacetate, which is involved in formation of citrates. And then after, citrate become decarboxylated to form 5 carbon compound and the carbon dioxide here, where 5 carbon compound is further decarboxylated to form another compounds, which we call 4 carbon compounds oxaloacetate. And ATP is formed in the process by substrate level phosphorylation. Now, the way of ATP formation in the process is by substrate level phosphorylation. Simply substrates having attached phosphate can transfer their third phosphate to that of ADP for carrying out the means of phosphorylation. Then after, the 4 carbon compound is oxaloacetate become regenerates the original 4 carbon compounds and react with that of another acetyl-CoA to start the process. Then after, the following product can be obtained after regeneration. Here there is 6 reduced NAD produced after regeneration from Krebs cycle, and again 4 carbon dioxide become decarboxylated too. And again there is 2 ATP molecule produced by substrate level phosphorylation. Additionally, 2 reduced FAD become produced here again. This is what the step of Krebs cycle looks like. Here 2 carbon compound is acetyl-CoA from that of link reaction, react with that of 4 carbon compound in fluid matrix of mitochondria in Krebs cycle, then form 6 carbon compounds called citrate. Then 1 carbon dioxide become decarboxylated, and with production of 1 reduced NAD. And again there is 5 carbon compound is produced. There is decarboxylation step here again, and again there is production of NAD takes place here again. There is phosphorylation takes place here again. Then 5 carbon compound become further decarboxylated into that of 4 carbon compounds, which produce FAD, and again further decarboxylation of this one. When this become regenerate itself to that of where this one become react with that of the original acetyl-CoA to regenerate the process again. Within 2 cycles, there is a production of these two products in Krebs cycle. So generally, in Krebs cycle, we end up with this 6 reduced NAD. We end up with that of decarboxylation of 4 carbon dioxide, and again we end up with the production of 2 ATP, net ATP again, and again with generation of 2 reduced fat again. Another most important step of aerobic respiration is oxidative level phosphorylation, where this oxidative level phosphorylation include both electron transport chain plus chemiosmosis. When we say electron transport chain, it is a means of transporting electron from one carrier to another. Electron can jump from one carrier to the next, whereas chemiosmosis means passing of proton through mitochondrial membrane. Here, the step of oxidative level phosphorylation totally forms these 2 reactions, which takes place only in mitochondrial membrane. The production of ATP takes place from that of ADP plus inorganic phosphate, which is the process of phosphorylation, whereas there is ATP synthase enzyme involved in the process. ATP synthase to form this ATP. Only ATP synthase takes place in mitochondrial inner membrane on the surface of crystal. Student, do you know the advantage of crystal during the process? Very good. Crystal is very important in increasing surface area of mitochondrial inner membrane to produce a great deal of ATP during this process. A great deal of ATP is produced by oxidative level phosphorylation because of the surface area of crystal, which increase surface area of mitochondrial inner membrane. The following reaction maybe takes place over the surface of crystal. Here, hydrogen atom carried by reduced NAD from Krebs cycle, and again carried by reduced fat from Krebs cycle, split into electron again, where the electron can pass from one carrier to another, which we call electron transport chain, making the system become efficient. Additionally, when the electron passes from one carrier to the next, they lose energy. The critical advantage of lost energies, making the system of electron transport chain to become more energetic, making the system of pump to become more energetic and powerful. When they are jumping from one carrier to another, there is a loss of energy. Energy loss is very important in powering the system, making them to become more energically jumping from one step to another, and highly enables the process to become accumulating the proton in mitochondrial inner membrane. Like that of the earlier reaction, there are 3 main electron carrier molecules that is found in mitochondrial inner membrane. One is reduced NAD dehydrogenase, which is act as a proton pump from one carrier to the next. And again, ubiquinone, which is important as a proton pump again. There is a cytochrome again, which is a proton that contain iron. These 3 are the 3 most important electron carriers that is found in electron transport chain. Well, at the end of electron transport chain, electron, which is split from that of hydrogen, combined with that of proton, which is split from that of hydrogen from earlier coenzyme again, binds with oxygen to form water molecule. So, in the process, oxygen is considered as a terminal electron acceptor of the respiration or that of the chain totally. Respiration ends with carbon dioxide plus that of water, where water is produced in this process. It is considered as a final electron acceptor, the terminal electron acceptor of the electron transport chain system. So here, as the action of proton pump, proton going to accumulate in where? In mitochondrial inner membrane space, where ATP synthesis is found. This is a reason for that of binding of ADP and that of third inorganic phosphate, which is a reason for that of phosphorylation. So here, there is a proton pump from outer mitochondrial space to that of inner mitochondrial space. Proton accumulation from outer mitochondrial membrane, which is fluid matrix, to that of inner mitochondrial membrane, which we call cristae, which is very important, and there is so many network of proton electron carrier found here, which is finally react with that of oxygen to form water. Finally, there is a formation of ATP here, where ATP synthesis is found here. So, ATP synthesis, the only enzyme that's found in where? Mitochondrial inner membrane, and again, chloroplast inner membrane. Here, when one NAD molecule become transfer proton from one carrier to another, it is a reason for that of passing of 6 proton from one carrier to that of the next. Additionally, when FAD molecule become passes proton from one carrier to another, it is a reason for that of passing of 4 proton through that of mitochondrial inner membrane, which is a reason for that of production of certain ATP. Let's see this one. An oxidation of one reduced NAD molecule results in 6 proton passing through that of ATP synthesis, which results in production of 3 ATP molecule. Hint again, student. When one reduced NAD molecule become oxidized, it passes 6 protons through ATP synthesis, which results in production of 3 ATP molecule. So, in oxidation of ATP molecule, which goes to that of ATP synthesis, we expect from 1, 3 ATP molecule in ATP synthesis. Again, oxidation of one reduced FAD molecule results in 4 proton passing through that of ATP synthesis. That results in production of 2 ATP molecule in ATP synthesis again, which I mean one reduced FAD molecule, which pass 4 protons through that of ATP synthesis and results 2 ATP molecule production. During glycolysis step, when glucose converts to that of 2 pyruvate molecule, there is 2 net gain of ATP by substrate level phosphorylation and 2 reduced NAD produced here, which is anaerobically takes place in cytoplasm or systole of the cell. Then there is a link reaction which generate acetyl-CoA. Then acetyl-CoA enter into that of a Krebs cycle, which can be a completion of all carbon in form of carbon dioxide, a completion of hydrogen in form of coenzyme takes place here. After regeneration here, there is 6 reduced NAD produced here, there is 2 FAD molecule produced here, there is 2 ATP molecule produced here. ATP production still here is by substrate level phosphorylation, where 2 of the ATP from that of glycolysis is important in shuttling process, proton shuttling. Then finally, in electron transport chain that involves oxidative level phosphorylation, there is about 34 ATP by oxidative level phosphorylation is generated here. Generally, from complete oxidation of glucose molecule, the net gain of ATP is about 36 ATP. Generally, student, In our day's lesson, we have seen some reactions that takes place in fluid matrix of mitochondria, which is Krebs cycle. During Krebs cycle, after regeneration of original oxalose state, we obtain 6 reduced NAD. We obtain again 2 reduced fat, 2 ATP by substrate level phosphorylation, and 4 carbon dioxide molecule. And then again, oxidative level phosphorylation, as it includes both chemiosmosis, a passing of protons through that of ATP synthase, and the electron transport chain, a jumping or transfer of electron from one carrier to another, which is a great reason for production of enormous ATP, which takes place in mitochondrial inner membrane. And again, we have seen when proton jumps from one carrier to another, they lose energy. The energy lost is very important, empowering, making the system of electron in the proton pump to become more energetic, and making the process to become accumulate proton in mitochondrial inner membrane. This is all about the today's lesson. In our coming lesson, we will see what happened to the respiration if there is no oxygen. Until that, goodbye, student.